let's talk about this lab today, because this lab is, I think it's interesting because it's taking the theoretical concepts and bringing it into a setting that's very clinically relevant. It's also really nice because it really makes you have to think about the concepts in very direct ways that, again, we are going to continue thinking about for the entire semester. Osmosis, osmolarity, and, things like that. It's like the more you can get just an intuitive feel for it, the better you're going to be doing. Is, when we get to the urinary system, what's going on in the kidney tubules. It's all about osmolarity and moving water, moving one place or another place, or so it's important to really have an intuition for these. And like, the more you spend time with them, the more you get intuition. Um,
I have a question for test tube 7.
uh-huh.
So urea crosses the cell membrane, right?
Correct.
So that means it will bring solution with it alone into the cell, or
Right, exactly. So we talked about, what is we can talk about that. We'll look at that one right now since you're mentioning it, but then we'll talk about the rest of them. So we have 0.3 molar urea. And we have, let's think about our red blood cell in here. Again, what is the osmolarity of the cytosol in the cell?
0.3.
It's also 0.3. 0.3 osmolar, and urea does not dissociate, so this is 0.3 osmolar as well. So this is an isoosmotic solution. It has the same osmolarity as inside our cell. And yet, again, urea it falls under the category that we were talking about. Although it's polar, it's small enough that it can penetrate. So if urea could not penetrate, then everything would be cool. We're balanced inside and out. But urea can penetrate. So urea starts coming in. And as soon as urea comes in, what's happening to the osmolarity in here?
It lyses.
It
starts to swell.
But so the osmolar, and it'll swell though, because we're adding particles. As we add more stuff in here, we're increasing the osmolarity. And now all of a sudden, it's higher osmolarity in here, and then the water is going to follow the solute, right? The water goes to where there's more solute. And so, yeah, it's going to start to swell. So hopefully, does this make sense? I know for some people it makes sense because people wrote kind of little eloquent things about it. So is there anybody who would like more clarification? This is an important point. Understanding why urea is going to end up being hypotonic, even though it's isoosmotic. Right? Basically, the water follows the solute.
I understand. So, but the answer will be is isosmotic, but at the same time is hypotonic.
Exactly.
Okay.
Right. So let's then just quickly review independent and dependent variable for our lab today. What is the independent variable? What are we varying?
Solutes.
So one is the type of solute, like sometimes we're using urea, sometimes we're using sodium chloride, sometimes we're using sucrose, sometimes it's ammonium chloride. So we're varying up what exactly the solution is made out of. And then what's the other thing we're varying?
The concentration.
Yeah, the concentrations. Sometimes it's 0.05%, 0.05 molar. Sometimes it's 0.1 molar. Sometimes it's 0.3 molar. Sometimes it's 0.15 molar. So we're varying two different things. What is our dependent variable?
What happens to the red blood cells?
Yeah, basically what happens to the red blood cell. And in particular, what's the thing that we can actually notice using our methods that we're using?
Whether or not it lyses or crenates.
Yeah, so we can check out, did hemolysis occur? If the supernatant is red, then we know the thing exploded. The crenation is going to be a little trickier, but if you have a comparison, you can make some assumptions about what's going on there. You won't have definitive evidence of crenation unless you actually look at it under the microscope and see them all shriveled up. You have definitive evidence of hemolysis if the supernatant stays red. So, and again, for the subset of you that still are having a challenge with constructing your objectives, objective is just going to be, how does the independent variable affect the dependent variable? Done. How is how does the concentration and type of solute of the solution affect what happens to the red blood cell in the, specifically, does it lyse or not? It's just it just has to, and there's different possibilities, but it has to just include the independent variables and how does that affect this? And then put a question mark after it, and then just, you're done. But again, it's an important part of any kind of scientific questioning is to have a very clearly defined question. So part of the reason I'm beating this dead horse, so to speak, is it really is an important skill to be able to really clearly and narrowly ask your question so you know exactly what you're looking for, how to go about finding it. And that is part of being a good scientist, is being able to ask a very well-defined, narrow question. So that's one of the skills I would like you to walk away from the semester with, is just kind of laser focus in what exactly am I interested in here and what do I need to do to get my answer. All right. I do apologize for the typo stuff. I had kind of spazzed on, first I spazzed on one thing, then I spazzed on other things. In the end, make sure you have the order of the tubes correct as you're keeping track of things in your lab notebook. Let's see here. I can zoom in even a little more. Right, so.
Sorry, I was just going to ask a question.
Yeah, what's your question?
It's kind of random, but I was just wondering for when we write the results for whether it's hemolysis or crenate, could we just say that possible crenation when we see that the pellet is a little bit smaller? Okay.
Exactly.
Sorry to interrupt.
That's good. So again, testing 1, 2, 3, 4, 5, 6, up to 9. Each of them needs a specific hypothesis. Again, most people did this, although some people didn't. And for each one, the core thing you need to know is the osmolarity compared to the cell, and a little more about, does it penetrate or not? And so for each one, that's all we need to do. So test tube 1, this distilled water. What's going to happen with that one?
It's a hypoosmotic solution, so it's going to create lysis or hemolysis.
Exactly. So this has an osmolarity of 0, right? There's nothing dissolved in there at all. So this is the most hypoosmotic you can get, pure 0 outside compared to 0.3 inside. So this is the most intense kind of hypotonic solution you can get, where the water is going to come in, and there'll be a lot of pressure. And this is almost for sure gonna pop. A little later, we're gonna, we would have been looking at them under the microscope. Instead, I'll show you photomicrographs of what they look like under the microscope. What are cells gonna look like? If you looked at this under the microscope after this hemolysis happens, what would you expect to see?
Remnants of the membranes.
Yeah, so maybe if you're lucky, you'll see little fragments, but in general, you don't see a lot, maybe a little bit of schmutz floating around. Again, imagine you're in a room full of these beautiful balloons on New Year's Eve, and the whole room is filled with balloons, and then they're all popped. And then you walk into the room and you look around, it's like, there ain't nothing here. And then you look on the floor, oh yeah, there's little bits of rubber around or whatever. But it's not that exciting to look at hemolyzed blood under the microscope. The fluid is kind of reddish because the hemoglobin spilled out, but you don't see much. Sometimes if they've hemolyzed very kind of gracefully and more deflated, you can get these things called red blood cell ghosts, which are kind of like just the membranes are still like deflated balloons. But in general, if you put red blood cells in distilled water and then try to find them under the microscope, you'd be just looking at little bits of whatever. Then we started getting the salt solution, NaCl, and we had 0.05, 0.1, 0.15, 0.3 molar. In each of these cases, what's the osmolarity going to be?
Two times the molarity?
Yeah, exactly. It's just going to be 2 times whatever the molarity is, because it dissociates into 2 different ions. This will become 0.1 osmolar. This will become 0.2 osmolar. This will become 0.3 osmolar. This will become 0.6 osmolar. So we got the osmolarity, and then we need to think about, does sodium chloride, is it penetrating or non-penetrating?
Non-penetrating.
Non-penetrating.
Penetrating. We talked about ions need channels to cross the membrane on their own. So if it's non-penetrating, that helps us think about this. What's going to happen in these different cases? What's going to happen in the case of the 0.1 osmolar?
Hemolysis.
Yeah, I'm going to expect hemolysis. I'm hypoosmotic. It's going to be hypotonic. Water will come in. What about here? 0.2 osmolar.
Same thing.
So the same thing. Is the osmotic pressure going to be as great as test tube 2 or test tube 1 where we had distilled water?
No, the concentration gradient is less.
Right, so notice the concentration gradient's less, which means that there's less osmotic pressure. You know, so if you think about a cell is not a, I mean, in the kind of language people have been using for the last few years, a cell is not a total snowflake. You know, as soon as, you know, there's a little buildup of pressure in or whatever, it doesn't necessarily completely explode. So you can have slightly hypotonic things that inflate, but don't necessarily pop, right? So the idea of being hypotonic, if it's more slightly hypotonic, it might not necessarily explode. Just kind of putting that out there. Or maybe some of the cells that are already weakened maybe will explode, but the cells that are healthier, that have more of an ability to adjust, will still stay intact. You know, I can say just from having done this same experiment for over 20 years now, we used to do the experiment with blood that was expired from a blood bank. So we would get it right after it expired from the blood bank, and we do the experiment. And I can say that the blood was always lysed in this situation. At some point, we shifted to where the instructors would give blood for the class. And then we found this particular solution typically did not explode the cells. You know, we realized it's just a healthy cell that just came out of your body actually has the ability to handle this slightly hypotonic situation. Whereas the blood that was already over a month old and was already no longer viable enough to transfuse into somebody, you know, you gave it one extra little thing like this and it all popped, right? So just kind of putting that out there, just because it's hypotonic, it does matter if it's like very hypotonic or slightly hypotonic in what happens. What about this one?
It's isotonic, so no change.
Yeah, exactly. It's isotonic because it's isoosmotic, the same osmolarity. And then again, it makes a difference that we know it's a non-penetrating solute. As opposed to the urea, which, you know, when I was talking about Amado's question there in the beginning, you know, there we had an isoosmotic solution of urea, but it was not isotonic because the penetrating solute comes in, the water follows the solute, the cell inflates and explodes. What about 0.6 molar or osmolar solution on the outside?
Hypertonic.
Yeah, hypertonic. Here we have way more salt outside than inside, so the water is going to move towards the solute out of the cell and shrivel up. And again, like I said, sometimes, especially when you can compare, like we know what the cells, if we have a tube where we know the cells are probably isotonic, meaning they're not shriveling or exploding, and then I can compare it, again, for the sake of, let's say there were 10 cells in here, and then I had crenation, they shriveled up, and I had 10 cells. They're gonna take up less space because they're, again, 10 raisins versus 10 grapes is gonna take less space, right? 10 prunes compared to 10 plums. They don't take up as much space. 10 little crenated blood cells compared to 10 happy, full blood cells. All right. And again, how would I know, just from looking at the tube after I've spun down, how would I know that they have all exploded?
If the tube is red.
The supernatant would be red.
And murky.
Why murky?
Just from the remnants of the membranes.
They're they're clear. You wouldn't really notice it. So not murky, but just kind of a clear. Actually, let me get a fat one here. A red supernatant. Would I have any kind of pellet at the bottom?
No.
Not really. If they all exploded, then there's no cells on the bottom. What about here? What if I had like partial hemolysis? I had some explode, but some of them actually didn't explode. They just got kind of fat. What might it look like?
Red tube with a little pellet.
Yeah, I might actually have the ones that exploded are going to spill out hemoglobin, but the ones that didn't explode end up getting pulled to the bottom as a pellet. So I might have some kind of a hybrid thing going on as well, right? So just when you're kind of thinking about kind of interpreting our results is, pure hemolysis, everything explodes. It's just going to be red supernatant, no pellet. Pure no hemolysis, I'll just have a pellet and a clear supernatant. But there's also partial hemolysis. Some of them explode, so I get some hemoglobin up into the water, but some of the cells stay intact and get pulled to the bottom when I'm doing the centrifuge. So that all makes sense. So then we got some other ones. We have tube 6, 7, 8, and 9. 6 is ammonium chloride. No, I did it again on you guys. Let me 0.15 molar ammonium chloride. Okay. Then we had 0.3 molar sucrose. We have 0.3 molar urea, 0.3 molar glycerol. So what are you going to expect for the 0.15 molar ammonium chloride?
Swelling or hemolysis,
Why is that?
because it's a penetrating solute. So it'll go into the cell and the water will follow.
So first, what is the osmolarity of this thing?
0.3.
0.3, yeah.
It's going to be 0.3 osmolar because it dissociates. So why do you think it should be a penetrating solute? Remember, in your hypothesis, you should have a reason. Like, why does it make sense that that ammonium chloride would penetrate? Right, because these are going to be ions. Right, ions typically don't penetrate.
On the first page of this lab manual, it was talking about chemical hemolysis. So in my hypothesis, I said that this solute will chemically destroy the plasma membrane.
Right, so that's a possibility. So I will say this is a particularly interesting one. But I want to make sure when people come up with their hypothesis that you have some reason. Like I will say in reality, due to a very weird thing, it actually can get across, but not in the way you think. Like just thinking about it as as an ionically bonded compound, it should not be able to cross the membrane. So like if I was originally making a hypothesis for this without having more information, I would say, I suspect this would be isotonic, right? Because it's kind of ions. The reality, we're going to talk more about when we get to the respiratory system later. It turns out red blood cells, when you're trying to carry, if you have CO2 in your plasma, the CO2 actually is carried mainly as bicarbonate ion. And the CO2 ends up coming in, there's a carbonic anhydrase in here, this enzyme carbonic anhydrase, and this becomes carbonic acid, H2CO3. This dissociates into H plus and HCO3 minus. And it turns out that as this leaves, that chloride comes in to keep the balance. And so there's actually this chloride bicarbonate exchange protein in a red blood cell to allow it to do this whole thing to transport carbon dioxide in your blood. And this little bicarbonate chloride exchanger in the red blood cell is going to interact with the ammonium chloride to allow ultimately the ammonium chloride to make it into the cell. But it's not an intuitive reason why ammonium chloride should be penetrating. It's only because of this special system that we haven't talked about yet. So just, in fact, one of the interesting things about ammonium chloride is it will selectively lyse the red blood cells, but not the white blood cells, because white blood cells don't have this bicarbonate chloride exchanger. So, it's very, it depends what kind of blood cell you're looking at, whether or not it's going to lyse or not. So just kind of putting that out there. Be careful about just, you want to have it turns out that it does penetrate, but you should have a reason why it makes sense it penetrates. All right, back here. So I would say this one is kind of a weird one. I put it in here because it is actually used to selectively lyse RBCs and not white blood cells. What about sucrose? What if I had 0.3 mole of sucrose?
Isotonic, so no change.
Yeah, so this is, we have isoosmotic, we have sucrose is going to be non-penetrating, so we have a nice isotonic situation. Urea, we've already just talked about. And again, urea is penetrating because it's small and it's polar, but it's really small. It penetrates enough that, the cells all are going to take on water and lyse. What about glycerol?
Would it also be isoosmotic but hypotonic?
So isoosmotic for sure. And why hypoosmotic?
Because it can also penetrate.
And why can't it penetrate?
I didn't get that far. I just found out that it could penetrate.
So you want to kind of think about why. What does glycerol look like? It's an alcohol. You know, and alcohols are kind of famous for kind of being able to like kind of have one foot in both worlds. You know, they're definitely got polar parts to them, but then they also, parts of it are polar, but parts of it are really non-polar. So they can like, they can hold kind of, they're kind of like a polar solvent and kind of like a non-polar solvent at the same time, right? Alcohol evaporates really fast because the molecules are really not holding together very tightly by hydrogen bonds. You saw that in Bio 110 lab. Remember that? You take water and you keep it in your glass and it stays in your glass. It evaporates really slowly. You have alcohol and it evaporates like that because those alcohol molecules are mainly, they don't have, the polar bonds are not all that, are not, nothing like in the water. So glycerol does have this kind of non-polar quality to kind of get into the membrane and cross, but it's not like a pure non-polar thing. And it turns out it depends on the animal. This is another thing when I talked about as doing this lab for a long time. When we used human cells, we always found the glycerol pretty much lysed everything. And then we started doing it with the cow blood. And it's like, whoa, the cells look happy. And so then we like went and looked deeper and found some paper looking at the relative permeability of glycerol across different species. And it turns out that the penetration of glycerol in human blood cells is much better than the penetration into cow blood cells. So what does that mean? So it still can penetrate cow blood cells, but it penetrates pretty slowly. So what is that going to mean in terms of what you're going to see when you put your cells into glycerol, spin them down, and check to see how the cells look, or if it lysed?
Maybe a hybrid?
Could be a hybrid. And what else is going to make a difference?
Maybe it'll take a long time.
Yeah, exactly. It's going to depends how long you let the cell sit in there before you spin it down, right? Like if it goes in, but kind of slowly, if you put the cells in and spin them down, they're all cool. If you put them in and they're sitting there and it's like, all right, I'm feeling more and more and more and more. Oh man, I popped. And then eventually you're spinning everything down. It's like, oh, I'm going to notice a bunch of lysed cells versus, right? Does that make sense? Right. So glycerol, particularly with the cow blood, it penetrates, but it penetrates pretty slowly. So we actually, for the procedure that you're going to see on the video, we actually added an extra tube. We added tube like 9, like with a star on it, which is the same thing. It's glycerol, but we let the cells sit in there for like a half an hour before we spun it down. So we can compare, like in your lab write-up, you're going to want to compare what do the cells look like if you just mix them in glycerol and then just spin them right down versus let them sit in glycerol for an hour and then spin them down, right? So
for the tonicity, could we just put hypotonic and isotonic? Like depending?
it's yeah, I would say yeah, it depends. I mean, at this point, you've already written down something, right? Hopefully this is going to be in your discussion, and whether or not it's going to be, it's probably hypotonic if you wait long enough is the idea. But yeah, hopefully, and I'm hoping most people have written up. Yeah, I mean, I know for you, sure, you've written yours up, so yours is fine. Just leave your hypothesis as it is, but then in your discussion, you can discuss what we actually see. So, so are there questions? And please ask, like, otherwise, like, I know from reading the pre-labs or from knowing some people didn't do the pre-labs, some people I think are pretty confused and other people are pretty solid on these concepts. If you have questions, please ask right now, because that will help you make sense of what we're going to see for this next part.
What are some possible sources of variability? The only one that I came up with was the type of a red blood cell. So human or like you were mentioning, cow.
So that makes a difference. The condition of the red blood cells as well. Like I was mentioning, the age, just the ones that have been sitting around longer tend to be less robust and more likely to pop under the same conditions. Other sources, what are other sources of variability on this one? Particularly in a lab, if we were doing this in person with 3 labs worth of students, so like 75 students all putting pipettes in the jars and putting it into test tubes. What is one of the real dangers of this?
Contamination.
Contamination. That's one that actually happens a lot, where somebody gets a little sloppy and puts the wrong pipette in the thing and then cross-contaminates across the different solutions. So contamination. There can be contamination in the test tube as well that you're using. There could be crap in there before you start. Other things like blood cells are heavy. Blood is a suspension. If you want to have a nice homogeneous blood drop to start, you got to make sure you mix up the blood before you take the little drops and put it into your test tube. That can be a source of variability. What are other things? I would say measurement errors. That's always one. Things like that.
Would you consider time one of them?
Not. Well, I mean, for glycerol, yeah. Actually, for glycerol, that is a source of variability. For this one that is slightly permeable, then again, if you let it sit for a long time before you check versus you just put the cells in the glycerol solution and spin them down, you get really different results. So yeah, no, actually, in our particular experiment with the cow blood and glycerol, it's a huge source of variability in what you're going to see there. In fact, we're turning it into a kind of secondary independent variable by actually controlling it and looking at how it affects the results in that case. Yeah, so there's probably more. Those are the ones that kind of come to the top of my head. All right. So
I have a quick question too, if I can ask. I'm still not like 100% clear on the ammonium chloride and how, just in general, we could tell if something is penetrating or non-penetrating. Like, what factors should we be looking at to be able to determine that?
All right, so that you kind of go back, when we go back to the phospholipid bilayer. The cell membrane. And then we talked about what can penetrate or not penetrate just based on the pure chemical properties. And things that were nonpolar, assuming they got up here, since this whole area, the membrane is basically hydrophobic tails. So nonpolar things, if they get to the membrane, can move to one side or the other without any trouble. So if something is nonpolar, it can penetrate. And again, glycerol is slightly nonpolar, which is kind of how it gets into this picture.
Okay.
we talked about things that are large and polar cannot, because, you know, H2O, heck. You know, so if this has, this is like, it wants to be attracted to the, it's not want to, it is attracted to the water molecules. So it gets up here, it says like no fun zone, right? There's nothing there that is going to be attracted to it. So it just stays on whatever side it's on. If it gets up towards this hydrophobic tail zone, there's nothing to play with. So it just is continually stays pulled on whatever side it's on. So large nonpolar things like sucrose, or like we said, the ions, something like sodium or chloride, because it has that sphere of hydration, all the water molecules go around it like a big snowball. It's effectively also like a large molecule, like a large polar molecule. The other things that can cross, we said were very small polar molecules, like H2O. H2O is polar, but it's so small that it's zipping around, it's going super fast. And it's like, whoa, I wasn't supposed to be in here, but before it even knows what happened, it's on the other side, like a bullet got shot through, right? It's like as the bullet enters into the membrane, it's like, whoa, I'm actually attracted back where I'm coming from, but I got my velocity, and before I know it, I'm on the other side, and now I'm happy over here. So small polar molecules can end up getting kind of thrown across just due to their translational, you know, kind of energy, and end up on the other side. So that's why osmosis happens, because the water can cross the membrane even though it's polar. And it's also how come urea can get in and across the membrane. It's has less permeability than water, but you know, it's permeable enough that it can cause it to be a hypotonic solution.
Okay.
So that's at the core of it. Like if you want theoretically, and then the other thing that is going to let you cross the membrane is if there is some specific membrane protein that allows it to happen, like for channels or carriers. And again, that's ultimately why the ammonium chloride is going to be able to cross, is because there's a special transport protein that is messing with things and allowing the chloride to move across and things like that. So that is the other thing. Things can cross the membrane if there is a specialized transport protein that allows it.
Okay. And so the glycerol, you said, has parts of the molecule that are polar and non-polar.
Right. So it has non-polar qualities, so that allows it to kind of get in there.
Okay, great. Okay, got it. Thank you.
It's more weak. It's not as attracted to the water as other things are. So it gets up here and it's like, yeah, I like the water, but you know, if I'm letting go of the water, I'm not, you know, it's, what would it be like? It would be like, you know, again, kind of like back to when I was talking about kind of like the big cuddle puddle and everybody like grabbing on and hugging each other, you know, water molecules with each other, or water and sugar molecules, they're all like bear hugs. Oh, I don't want to ever let go. And like the glycerol is like, oh, nice to see you. Let me move on. You know, it's like they're attracted, but it's not, they're not giving a bear hug. So it's more likely that they could end up getting kicked across the membrane because they're not so yanking onto the water molecules on either side. That makes sense.
Yeah. Okay, got it.
Okay. Okie dokie. So Are there other questions before we actually just get into the actual data? Okay, so we're going to be doing, there's 3 parts to the lab. One is going to be the test tubes. We're going to spend like maybe the next half an hour or so just kind of going over, looking at the actual results. The module includes pictures and stuff, so don't worry about, I'd say for the first part, for right now, just kind of follow along with the videos. There are like photographs of what the tubes look like that you can then use to put in your lab notebook, and that you can look at more in detail for actually describing what happens in each tube, and whether your hypothesis is confirmed or not. So this is going to be the first part, is the test tubes. We're also going to be look at blood cells under the microscope. I mean, normally, if we were in lab, we would actually take little samples out of the actual test tubes we've been using and look at them under the microscope, so you can get a sense of blood cells, get a sense of crenation, this and that. Instead, I'll go with you through some photomicrographs so you can get a sense of what cells look like under these different conditions. But then you should, like in your write-up, you'll have to include that, and include so you have a sense of not just what does the test tube look like and the supernatant and the pellet, but then a deeper level, what do the cells look like inside there as well, in terms of, so we'll do that. And 3, we're going to do like this, the cell model, kind of dialysis tubing sausage. Again, usually if we're in person, particularly I like this because it's hands-on. You can poke at it and stuff like that, because cells you're doing more in your brain. It's like, it's all kind of theoretical, and even if you're seeing things, you're seeing them under a microscope, or, this is kind of nice because it's some physical thing you can play with. Unfortunately, we're not in lab, so you're not going to physically play with it, but you'll get to watch Tina play with it. And hopefully that will give you a sense. Again, another level of hopefully intuition about hypotonic, hypertonic, isotonic, because those concepts are really deep in our class. And I really want you to just keep seeing it again and again and get this intuitive feel for it. So we'll start with the test tubes. Okay. Let me get my, right. Let me not get into trouble here, because I know, let's get the, okay. Share screen. Okay. Okay, so fluid balance and RBCs, pre-lab. This copy of the lab manual pages, which is this PDF, this is the, if you want a copy of the instructions that actually have the correct order and correct concentrations, you can just print out page 2 of this. This actually, or you could just scribble it out and pay, on the one you have. Either way, either correct the molarities on the typo in your lab manual that you bought, or you can print this page and use this one. It doesn't matter. But make sure that your procedure in your lab notebook reflects the correct the correct molarities and stuff. Wait, then where do I gotta go to the next page. Back. Oh, it's over here. Okay, next. So hopefully everybody watched this introduction. That's kind of where and this is, so for those of you who did not do the hypotheses for each thing, please try to read closer. It talks about, make sure you have a specific hypothesis for each, capitalize of the 9 tubes in the lab. And then this is talking about, I have this little video here. So I try to give you the background so you can work with this stuff. And again, a lot of people did kind of bang up jobs on this, but some people really dropped the ball. So if you're one of them, you really do want to kind of up your game a little bit. And I included this table here. We can look at it later, but kind of giving you a sense of what you should think about. What does it dissociate into? How do you calculate osmolarities from molarity? All right, and then people did this. Okay. Doing the fluid balance in RBC's lab. And here you can see I actually have pictures of what, and we'll look at these in a few moments. But for right now, let's go watch a scintillating video. So I'm gonna start. Transport across membranes, fluid balance in RBCs, setting up the tubes for part 1, starring me. Yeah, this is always kind of embarrassing. With 3 mils of distilled water in test tube 1. Again, we've already discussed the proper use of these pipettes. I've sucked up to the 0 here, putting it in the appropriate test tube. Now I dispense down to the 1 mils dispensed, 2 mils dispensed, 3 mils dispensed, and that's that. I'm going to get 3 mils of my 0.05 molar NaCl. Again, just suck up to the 0 and put that in the appropriate test tube. Dispense 1, 2, 3 mils. Notice it's going to be important for you to be precise with the volumes. One, just because we want to make sure all the conditions are controlled and as similar as possible, except for our dependent variables, which we're changing, but also because we need to make sure things are balanced in the centrifuge. And think about if your washing machine or your dryer is off balance, there's more weight on one side or another, it goes boom, 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 boom. It's even worse if you're going super, super fast, high-speed centrifuge, and we don't want any disasters. And you need to make sure Tina will talk more about the balancing, but part of the balancing is making sure each of your tubes is got the same volume. So what I'm going to do is I'm going to put 0.3 mils of each of the appropriate solutions into their appropriate test tubes, and then we will come back in a moment. All right, so we have each of the test tubes with the appropriate solution. And now each test tube is going to get 6 drops of the blood. Like I said, make sure it is well mixed, because the suspension, you're going to have the cells falling out. And again, because we want to only vary the solutions, we want to make sure the amount of blood in each test tube is the same. So these drops, we try to make them as consistent as possible. It's going to be 6 drops. And it's not just around 6. I really want to do 6 well-formed drops. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. One, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Should also mention, this is cow blood, so we don't worry about the safety as much. But if this was human blood, obviously we would be very concerned about safety and be using universal protocol, universal precautions and all that. Let's see. Another 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Notice I'm also refilling before it's empty, so I don't get into a weird position where I've done part of the drops and get foam coming out, and then it's not a well-formed drop, and it's like, dough. So trying to preemptively make sure I get 6 well-formed drops. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. I can also mention we have one tube here that we actually put the blood in about a half hour ago. This is a different version of tube 9 with the glycerol. We'll talk a little more about why we've created this, but we're going to be able to compare the blood that's been in glycerol for here over a half an hour versus the blood that we've just added to glycerol just now, which we're going to be spinning down presently. So we'll be able to see how the amount of time the blood cells have been sitting in the solution affects the results. So again, we actually have 2 versions of 9. And the next step is going to be, make sure these are kind of well mixed. If they don't look well mixed, you can just use like a clean, this is a clean pipette, and just kind of really make sure they're all mixed up well. And then obviously you're not going to use this for anything else anymore because it's going to cross-contaminate. So this is, plastic waste. Hopefully it doesn't poke the nose of a turtle in the ocean or something. All right, that is that. So next we will get a tour of the centrifuge. Right. So I put that in there just because I do want people to be aware that we are conscious of. I mean, everybody's seen that horrible video of pulling the straw out of the turtle. I mean, unfortunately, medical waste is one of the huge places where there's single-use plastics. And so trying to just, be aware that we need to use plastics sometimes because they make sense. But my public service announcement is reduce use of single-use plastics whenever possible, because there's lots of times we use them when there's no reason to use them at all. And they're really messed up in the environment. All right, continuing on. So I'm going to start with 3 mils of distilled water in test tube 1. Again, we've already discussed the proper use of these pipettes. I've sucked up to the 0. Wait. Okay. Different version of tube 9 with the glycerol. All right, so any questions about the setup of everything? It makes sense. We just took the different solutions, put the blood, exact same amount of blood in every one. And again, it's important that the volumes are all the same, because when you spin things in a centrifuge, if it's off, you're going to have the thing doing this, minimally walking. Worst case, it's going to like fall off, or you don't want to have it like fly apart and get shrapnel or whatever. So that's the setup. And now we're going to have the centrifuge and the results. So let's spin it down with your host, Tina.
All right, so next we're going to centrifuge these test tubes. Unfortunately, we have more test tubes than we can finish in one round. But let's start off with how to use the centrifuge. So number 1, make sure it's plugged in. Number two, on the back side here of the centrifuge, you have a switch. And so now it's on right there. And then once it's on, the display will light up. And it should already be set on the settings that you need. So we're going to spin these for 5 minutes and 5,000 RPM. So you might go, well, it's only saying 500, but if you look up here, it says times 10. So 500 times 10 is 5,000, so this is good to go. RPM means revolutions per minute. So that means that it's spinning 5,000 times per minute. That's quite amazing. So we're going to get a lot of G's, and it's going to separate out the components of this fluid based on density. And so we're going to get the more denser particles at the bottom. So our red blood cells will be down there forming a pellet, hopefully. All right, so next we're going to press open. And if we look inside, there are slots here that are numbered. But as David mentioned before, you should label the test tubes, because once again, we're gonna have to do this twice. And so just being organized from the beginning is kind of key. So next, I'm just gonna make sure once again that everything is balanced, because otherwise this thing will not be happy. And so,
and that's another, remember I talk about like those, you double check yourself, because your people are imperfect. You pipette and you mess up. And so it's good to do like double checks, like before you put them in, they should all be the same height. And every once in a while they're not, in which case you're like, oh, I'm glad I checked. So, and that's another thing I think of this lab is just realizing like, it's okay not to be perfect. And that's why you need to kind of double check yourself on things.
Just stick things right across. So if I were to only do 2 tubes, now this would be balanced. But then I just keep adding tubes across from each other. As long as they are the same volume, they have the same weight, and everything should be fine. And I do not need to cover the test tubes with any parafilm or anything like that, because you can see that they're slanted, they're angled out, and we're going to have the centrifugal force moving everything out and downward. So there should not be any risk of anything spilling over. All right, so we're now set. We're going to have to save 2 test tubes for later. You close it, and then all you need to do is hit start. And this thing should start up, and then it stops automatically after 5 minutes. All right, so the centrifuge has just finished. I can hear it kind of slow down, and you can also look on the numbers that it's slowing down right now. It says 62, 58 RPM, and so you can really, see that it's slowing down. The lid probably won't open until it's completely still, because you don't want to lose any fingers. There we go. It's also giving a nice little beep. So anyway, I'm just going to take these out. And since I have them numbered, I don't need to pay attention to exactly where they're at now. It's more important for me to get the next batch started. And so I can rearrange these later. So next, you guys would only have number 9 left, which is the glycerol. But remember that we're doing a little special experiment now where we left another glycerol in for a much longer time. And so it turns out I have to balance the centrifuge. But for you guys, you would only have this one. And so what you need to do is create a dummy tube that weighs the same. And so what I done here is I measured out 3 mL of just water, and then I added 6 drops of water to make sure that they're exactly the same. And so you would just kind of put them right across from each other and do the whole process. So in this case, since I do have our special number 9, I'm going to stick it here, and they're right across each other, and we can start this centrifuge up one more time.
Right, so again, one of them has just been in glycerol for 5 minutes, because it was waiting while the other ones were spinning. but 9 has been, at this point, it's been almost like an hour that it's been kind of sitting, the blood cells have been sitting in the glycerol solution.
And we'll come back in 5 minutes and take a look at that. Now I'm gonna start taking a look at what's happened in my other tubes. So we have 1, 2, 3, 4, 5, 6, 7, and 8. And so if we just kind of do a visual look of these, you clearly see number 4 and 5, they have clear supernatant. So the fluid on top of the pellet is known as supernatant. And so those have clear. You also see it in test tube 7. And then all the other ones have some shades of pink. And you can see a little bit of a pellet down at the bottom here too. So we'll wait until we have the next batch too, so we can do our full comparison. We're gonna have to take a look at some of these pellets under the microscope to actually figure out if they cremated or if they're just the same as they were before. Because remember, they get pink when the cells actually lyses and we get the hemoglobin out there. But when they cremate or they stay the same as they were from the beginning, that's not going to be indicated by your supernatant. And so in those cases, we most certainly should break out the microscope and take a look at them and see if they're actually swelled up, or sorry, shrunken up or are the same. All right, we'll come back in a few.
All right, so we finished spinning down the last samples here. Now we can look a little closer at what happened. You know, again, the one that was in distilled water, you know, basically osmolarity 0. This is the most hypotonic you can get. We can see the supernatant is bright red. And if you look for a pellet, it's really not a pellet, which is telling us that all the cells are popped. All we got are little shreds of membrane in there. And in fact, a little clue, when we look at this under the microscope and you're trying to see what happened, like, you know, imagine you were in a room full of balloons and then every balloon got popped. And then you walk in the room and like, let me see what it looks like in this room full of popped balloons. It's like, it's not going to look like much of anything. Maybe you're going to see some shreds of rubber on the floor. So people get kind of frustrated when they're trying to like, what am I supposed to see here? You know, think about that. And again, we have varying hypotonic solutions. And one of the things that's interesting is we go to the 0.1 osmolar, 0.2 osmolar. And the 0.2, even though these are both hypotonic, we have red, which tells us we had lysis going on, hemolysis. Notice the pellets are actually different. Here, not all the cells necessarily popped. This test tube 3, this one with 0.2 osmolar, was 0.1 molar NaCl. As we've done this lab over the years, we actually get different results, actually depending on the freshness of the blood. Blood that is fresher, the cells are healthier, more robust, they can actually take a slightly hypotonic solution and they're gonna puff out, but not necessarily pop. Whereas the older cells are not able to sustain that kind of increased pressure and are more likely to explode and create this hemolysis condition. So, what you get in this tube might actually depend on just how old or how healthy the blood cells are. And we can see the one that is in the 0.3 osmolar solution. This is what we would hopefully expect the cells to be in an isoosmotic, isotonic solution. And we see in general, the cells all just got pulled to the bottom intact. There is a slight blush here, which you wouldn't really expect, except I think in this case, the cells have been sitting around for a while. They might be just some cells died off just because they're getting old. not necessarily because of the solution did it to them. This is the 0.6 osmolar solution, the really hypertonic solution. And here we see also the cells are intact, no cells exploded, but I don't know if you can see from your, can you see from your point of view there? Like I can see from my point of view, like a pretty different size and pellets. You know, as when I was talking about the raisins, think about the grapes. These both have exactly 6 drops of blood, the exact same number of blood cells in them, but the amount of space that those blood cells are taking is different. And this is our first clue that these cells here probably crenated compared to these because they're not taking up as much space for the same number of cells. And we're going to be able to follow up on that when we take them and look at them under the microscope. You know, and obviously a crenated cell looks very different. It actually looks all shriveled up and spiky. You can see the cytoskeletal elements poking out against the membrane. They look kind of like, I don't know, pufferfish or something. All right, so test tube 6, this is the ammonium chloride. You can see we have no pellet. All the cells seem to be lysed, and you know, the hemoglobin dissolved in the supernatant. This one's a tricky one. We'll talk a little more later about what happened here. This one's not so intuitive. It very likely did not follow your hypothesis, which again is all right. Hypothesis does not have to be correct. 7, this is going to be the 0.3 molar, which is also 0.3 osmolar sucrose. And we can see a really nice well-formed pellet with a relatively clear supernatant. Here we have the urea, and we can see the urea is also got a red supernatant and really no pellet. So we're assuming here most of the cells popped. And then finally we have the glycerol, and we have 2 different conditions here. Can start with this one here. This is the glycerol where the cells were only in for a few minutes before we spun them down. And we can see some of the cells lysed. There's red here. That means some of the hemoglobin escaped into the solution. But there's a pretty well-formed pellet, which is telling us a lot of the cells actually did not break apart and then got pulled down, complete down to the bottom of the tube. Whereas in this one, where the cells sat in the glycerol for about an hour before we actually spun them down, there's no pellet, which is telling us that the longer you sit in the glycerol, the more likely you are to explode. And we're going to talk about that more in detail as well. So kind of why that happened and why that makes sense. And actually we'll even talk about how this part would be different with human blood cells versus bovine blood cells. So we'll also provide pictures of each of these tubes as well for you, so you can refer to that as well when you're writing up your discussions about results and discussions about what happened here.
All right, so next we're gonna.
So obviously we've already discussed all that stuff. And again, so in your lab write-up, you know, you can go back at that. And again, I have pictures here. I've got the, oops. Oh my gosh, hold on, what happened here? Why isn't it moving? Oh, you can see it. No, so these, yeah, so just use these. You can see up close, you can see the difference between 2 and 3. You know, 1 and 2 total hemolysis because red supernatant without any kind of pellet, whereas 3, you can see this partial hemolysis that some of the little blood cells survived by making it down to the bottom. 4 and 5, you know, no hemolysis, However, the pellet's a little smaller in 5. 7, no hemolysis. 8, here again, the 9 and 9 star, you can see a big difference. Both there's some hemolysis, but in the one that was only in for 5 minutes, a lot of the cells survived. Whereas the one that was in for over 30 minutes, they all exploded. This is kind of showing in more detail. It's kind of blurry, but it still shows. This is supposed to be the 0.3 versus the 0.6 osmolar NaCl. I should probably, it's basically these tubes 4 and 5 here. You're looking at the detail of the base there. I should probably edit this to make sure you realize this is tubes 4 and 5. So here you can really get a sense of the pellet being smaller in the crenated condition than in the isotonic condition. So are there any questions about the results of that part? Okay. And did we lose is Paige here? Or Juliana? Okay. All right. All right. So the next part, normally what we would do is take some samples. We'd resuspend the blood a little bit and we'd look at them under the microscope so you could get a sense of what they actually look like. I'm going to show you this little video which just shows the process of making the wet mount, which is something you've also, if you're in micro, you do as well. And it's worth, it's a skill. Again, normally you'd actually get it in person, but it's worth at least seeing if you haven't seen it, like how to make a wet mount to seal the edges so the stuff doesn't evaporate. Because if you do if you just put a slide on top or cover slip on top of your slide, you know, stuff evaporates pretty quick and changes what it looks like. Well, actually, hold on, let me look at the time. Let's take a break. Let's take like a 10-minute break so you can a bathroom break or whatever. And let's say at 11:45, we will come back and let's finish up this lab. So let's take a break till 11:45 and then we will do the photomicrographs and the sausages.
Professor, quick question for you.
Sure.
So for my hypothesis, I had just filled out that form that you had. So I'm going back through it just to revise it and resubmit it for you. Would you like me to just number out each test tube aside from that worksheet that you had, and give a written hypothesis for each one, or would that satisfy?
You need to at least minimally, you should mention like how, like if you just put like hypotonic with P with, you should at least be more explicit, like how those relate to create your prediction.
Understood.
So I would say, and I don't remember, so as long as there's as long as it's clear that you've thought it through, and then why, that you could just put even a few words like on the side of the chart or something, or you could write a separate thing. There's no right answer. Yeah, I'd say.
Okay, yeah, I've just done a revised hypothesis altogether, so I'll just write a sentence about why it is, but I'll include that because I already put that in my lab book, that worksheet that you had. So
yeah, that sounds perfect.
Okay, thank you.
Sure. Okie dokie. All right, so what we're gonna do now is watch a little video on making the blood slides, and then we'll just look at some images of what the blood actually looks like under the microscope after it's been in these different solutions. So just so you get more of a real sense of what is happening to the cells inside the test tubes. So here we go. I'm going to share screen. All right, so if I want to actually see if my cells truly created, the only way to really see that is to look at them under the microscope. So I'm going to talk a little bit about how to prepare a microscope slide to look at the blood. You know, so let's say this is test tube 5 where I'm assuming they created. First thing you're going to need to do is resuspend the blood cells up in the solution, obviously. You don't want to totally mix it back up. If you put too concentrated blood cells on a slide, it's hard to see any single blood cell. It's like trying, if you're interested in looking at a grain of sand, but you're looking at a whole handful of sand, all you're going to see is just crazy sand grains everywhere. So you want to just suspend a few of them. So don't get too overly ambitious about resuspending them. You just kind of get a few back up in the solution there, and then you can grab them up, but you really want it to be pretty darn dilute, or else it's going to be really hard to see any individual cells. And then we want to actually create the actual slide. All right, so to make a wet mount of the blood to see what happened to it, we want to make it so the cover slip is sealed, so the water doesn't evaporate from the solution if it just sits there. And so there's a nice little technique to make a sealed wet mount using Vaseline. You start, this is just regular old petroleum jelly, Vaseline. You take a bit and you make a thin layer across this webbing between your thumb and your forefinger. You know, it should be thin but present. Then I am now going to take my cover slip, and this is just very thin glass, and I'm always going to make sure I hold it just by the edges because I don't want to get my fingerprints all over it. And I'm going to, for each of the 4 sides, I'm going to put the side into the Vaseline and just drag it. Maybe it's like about 1/4 of an inch, and it brings, I don't know if you can see that, there is, the Vaseline has created a seal on that one side. And then for 90 degrees, go again. Now I have Vaseline on another side. Rotate it again 90 degrees. And Vaseline on 3 of the 4 sides. Again, 90 degrees. And I'm running out there. That's good. So here you have Vaseline on all 4 sides, kind of underneath, and it's going to make a nice gasket. When I put this down on top of my main slide, it's going to seal around the edges, and I can show you how that works right now. So here is my slide. I'm going to take a drop of my resuspended blood cells here. And, that's probably plenty. I'm going to take my slide and start one side, drop it down. I'm going to use the back of my fingernail to push it down a little bit. And what we have now is a sealed wet mount. The blood suspended in solution is sealed inside the Vaseline underneath there. So this can be sitting around now for like a half an hour and it's not going to change. If you did not do, it's a bit of a production to actually do this sealing, but if you didn't, like 10 minutes from now, you start having like salt crystals probably under there as the water evaporates and leaves into the room. So this allows you to have a nice slide that you can come back to and show other people as well. And this is what you would do for a variety. We talked about in the lab manual. Look at test tube 1, look at test tube 3, look at test tube 4, look at test tube 5. And so you do the same process for each of those, and compare and draw. For our online version, we'll show you pictures. But this is the only way you really know what really happened to the blood cells, particularly if they didn't lyse. So the what now of the blood. What? WandaVision. If you're not watching WandaVision on Disney Plus, you are missing on the TV sensation of the century. Whatever. All right. Here we go. So if you go to the next page in this module, I have some just photo micrographs of what you would actually see. So like in your lab write-up, I would say it's not a bad idea to maybe print some of this out and paste it in there so you have a sense of what they look like. And then, in your lab, it asks, you look at these under the microscope, write down like which one of these would you expect to see? Like for normal RBCs, isotonic solution, which test tubes would we expect the red blood cells to look like this or like this?
Urea.
Urea. What do we expect the cells to look like in urea? What happened in urea?
Hemolysis.
Yeah, hemolysis. So this is not hemolysis. This is the cells are happy. This is what a normal red blood cell looks like. So it's definitely not going to be urea.
So that might
It's
be
an isomonic solution. So
Yeah.
like a OT3 of the NaCl.
this tube 4.
Yeah, so it'd be like the 0.15 molar NaCl or the 0.3 molar sucrose, right? So in a red blood cell, we'll talk way more about them, but they eject their nucleus when they are mature. So they their cheeks dimple in because the nucleus pops out. So they're kind of round, but then the sides are sucked in. So this is, when you see a red blood cell, it always has this kind of dimpled look because it's round, but then the nucleus is ejected. So the sides suck in like that. So these are happy red blood cells. These are what you'd find in an isotonic solution. So, make sure you think about isotonic happy, they look like this. Here you can see what happened to these cells.
Did they shrink?
Yeah, they shrink. Take a look compare this to this little spiky dealy bopper. Here you can also see, like I said, the little cytoskeletal elements kind of poke out. So I always think they look like little puffer fish or something, but they get all spiky and small. What test tube would they probably look like this in?
Test tube 5.
Yeah, exactly. The one where you had the hypertonic saline, right? So this is what happens when you have a little cell that loses its water and shrivels up. These are like the raisins I'm talking about. Right, so we can, let me go back here. You know, in this page here, you can see they're isotonic, although every once in a while you see ones that are looking a little like starred up. Here you can see hypertonic, where most of the little cells look like little pufferfish, like little spiky raisins. Again, hypotonic, but slightly hypotonic. Kind of compare the cells that are normal, where you can have that dimple. That's a normal red blood cell. Again, all these things, you can see a normal red blood cell has that kind of, both sides are sucked in. If we have a slightly hypotonic solution, what are we going to expect?
Swelling.
It's swollen. So they're swollen. So they're kind of, you know, they're inflated. They don't have their dimples anymore, but they're not exploded, right? So they're still existing. So which tube might we find this in? Like ones that are inflated, but not necessarily exploded.
Tube 3, the 0.1 molar sodium chloride.
Exactly. Those ones that were sitting at the bottom of our tube 3, the ones that they're probably inflated, but they did not actually explode. So they probably look kind of more like this, kind of more like an overstuffed red blood cell. So just kind of giving you a sense, you know, you can have normal, crenated, if it's hypertonic, hypotonic. If it's slightly hypotonic, they'll be inflated. If it's very hypotonic, what are you going to see? Like tube.
Hemolysis.
Yeah. So what would this look like under the microscope for like tube 1?
Clear.
It'd be pretty much clear. You wouldn't see much of anything. You know, again, maybe you'd see some little fragments of stuff, but in general, you would look for something, you wouldn't see it. Kind of like my analogy of walking into the room, looking, scanning around for the balloons, but you don't see them because they're all just popped. So there are questions about this. Like in your lab notebook, you should connect what the cells look like to the tubes that we talk about in the procedure. I do want you, even though you're not going to look at them under the microscope, you're not going to draw what you see. You can, you know, print out and put a picture from one of these, what you would have expected to see in that tube. I do want you to connect the actual cells with what's happening in the tube. So any questions about that? All right. Let's see. All right, so there's only one last part to this lab. So I'm going to go back to the previous. So like I said, one of the things that we like to do in this lab is give you a larger macroscopic example of, you know, cells in a hypertonic isotonic solution that you can actually poke at with a stick. But instead, you're going to watch Tina poke at them with a stick. But in your lab notebook, you should look at this video and you should answer the questions. Describe which one is which. So I'm going to show you this little video, but again, it's 3 minutes, and you can go back and check it out as well. Does everyone remember what dialysis tubing is, or dialysis membrane? Right. So remember, we're going to fill these with sugar water. If I have a dialysis membrane sausage filled with sugar water and I put it in distilled water, what would happen? Right? If I had a little tube made out of dialysis membrane and I tied off the ends, let's say it was filled with 1.5 molar sucrose, and I put it in just a bowl of distilled water, what would happen?
It would swell or pop.
Yeah, so it would swell, right? Because we have a hypotonic solution. Water is going to come in and it would swell. And whether it popped would depend on how strong the plastic is, and how tight the clips are on the edge. When we do this in lab, I actually do have to be careful. If I leave that in the water too long, they literally do explode. But the plastic is strong enough that it can hold on for a couple of hours before it fails catastrophically. So here, so watch this and you can see the tubes in action. Okay.
All right, so we're learning about osmosis and tonicity in this course, and so here we're going to do a demonstration of osmosis. So what I have here are 4 kind of sausages made by dialysis tubing. So we fill them with 1.5 molar sucrose solution, and so all 4 have the same, and they're about the same size. Next, we're going to stick them into different solutions, and we're going to see which way the water is going to move. As you remember, osmosis, we're going to see the water is going to move from a low solute concentration to a high solute concentration. Or another catchy way to remember that is that water chases solutes. So I'm going to stick each of these in here to distilled water. I'm going to put one in here to 1.5 molar sucrose solution, which is the same as the inside of the sausage. Remember, all of the sausages have 1.5 molar sucrose solution. And then this last one has 3.0 molar sucrose solution. If I stir it, you can see it's quite, concentrated and sticky and thick versus here it's still has sugar in it, but it's a lot more dilute. And then distilled water, of course, is just distilled water. I'm also going to leave this control sausage here because when I come back in 30 minutes, I can tap on this and kind of feel what was the original kind of, consistency of this sausage, and then compare what has happened to the other 3 sausages. So we'll come back in 30 minutes and take a look at that. All right, so we're back at our sausages here made out of dialysis tube, and it's been actually an hour. And so just to kind of check on these, here's our control. So you can see it's, kind of this consistency here. I can lift it a little bit of bending, but, we filled it with sucrose solution. This has been in distilled water, and if I push on it and also lift it, you can see it's very rigid. So this one has taken in water and got quite taut and rigid. This one has been in the same concentration as we put inside the tube. And so this one is very much like the control sausage, kind of same consistency. And then if we go to the one that's been put in a more concentrated solution, you can see that it's pretty soft and flaccid. So obviously this sausage has lost water to the outside solution. All right. Now it's up to you to figure out exactly what happened, which way did water move, and what do we call the solution that we put them into.
All right. Right, so I mean, that's pretty straightforward, but it's still, again, wanting you to be thinking about these concepts from lots of different angles. Just there's a little chart in your lab book. Answer the questions like what happened to the water? What do we call the solution? And again, part of like looking at that, you get to just see this thing that's big and what happens to it. And I should mention, just to be totally honest with you, that solution that we call 3 molar sucrose, it's actually not exact. It's more like 3 molar sucrose wouldn't be quite that goopy, but I like using a really super goopy sugar solution because it really kind of drives home, this is really sugary, right? So just so you get a sense of like lots of sugar, like in a very kind of visceral, tactile way when you're poking at things. So if you're making 3 molar sucrose solution at home, it's not going to be quite like that. I actually just poured some Caro into there. But it works. It does a good job of being, what would I call that solution compared to the sausage? Of all the different terms we're using, this blancotonic solution.
Hypotonic?
Hyper.
Yeah, Tim, yeah, it'd be, if it's super sugary outside compared to inside, that would be a hypertonic solution, right? Hypotonic, which one of these would be a hypotonic solution?
Distilled water.
Exactly, the distilled water. That's where it's a lot less than inside the tubing, right? So again, this is just another place for you to keep working with these basic ideas, both thinking about it in the cells and then also just, how is the dialysis tubing sausage different from an actual cell? How is the membrane different from a cell membrane?
Doesn't a dialysis membrane only allow water to move across?
It can allow other things, although it's very non-subtle. It's just based on the size of the pores in the plastic. In fact, the word dialysis is kind of about movement of stuff as well. So there are things that can move across that membrane, but not sugar molecules. But it's much more mechanical. A real cell membrane, hopefully you're getting a sense from our talking about cells, is that they're much more dynamic. They have gated channels, things that are open, sometimes closed at another time. This piece of plastic is not nearly as interesting. It lets water through, it blocks sugar. It never lets sugar through. Whereas your cell, sometimes your cell lets sugar through if it wants, and sometimes it doesn't. Sometimes it actually actively pumps sugar across the membrane, right? Just kind of give you a sense that your actual cells with all of their membrane proteins are very dynamic in terms of their permeability. Whereas this dialysis membrane is very static. It's just kind of defined by its mechanical properties, and that's it. So just kind of, kind of putting that out there. It's thinking of cells as these really dynamic systems where lots of stuff is going on and they're changing their properties depending on what's going on around and what the necessities are for their current their current state of what's around them and what they're trying to do. I think it's important. Hold on, one more thing then. So let's see. No, this isn't it. So hold on. Give me 2 more seconds here.
I would like very much
remain on. Little the toxic
Well, I want to.
it's axial resonance.
Hold on 1 second.
Twinkle, twinkle. And that's the typical way it works.
Hold on. See. Let me see if this works.
With each new this combination has led to microscope.
Okay.
And then induce these to emit fluorescence using a stripe pattern at a second wavelength. This combination
in particular, you may see full of actin and growth plays of high resolution.
No, I'm not finding the actual video I want to see. There's this guy Eric Betzig. He's fascinating. He came up with these really amazing ways to image inside the cell. Videos of cell organelles. Let's see. You know, for the next class, I'll find you some good videos. I went to a talk by him, and he just showed these insane videos of how dynamic. Because before we kind of leave, we're going to be leaving the cell soon and starting to put cells together and having cells talking to each other's cells. But I do want you to really kind of get a sense of a cell as just kind of crazy busy on its own. Like, you don't need to put cells together to make things interesting. You can look at just a single cell, and there's so much going on in there, and it's so active, and there's so many different organelles all interacting with each other and moving around and altering and changing. So let's just leave that there. So the other thing I should probably say, the reality is the stuff I want to have covered for exam 1, I don't think we're not going to be able to cover in one lecture on Thursday. And yes, I think I'm probably going to move exam 1 to the next Tuesday. That gives me a little more wiggle room so I can cover the stuff, finish covering it on Tuesday, and not feel like I'm just like, because I could just lecture over our lab and just have like a 4-hour lecture, but then that's kind of mean. So I think what I'll probably do is move exam 1 to March 2nd. So I will send a announcement updating that, and I'll update the schedule on Canvas. But I think that makes sense. So we can cover, because again, it took me, I'm still figuring out how long it takes to cover things in Zoom as opposed to just being in front of a board. So I'm a little behind compared to where I thought I might be. So are there any questions about the lab or about osmosis or anything like that before we kind of call it a day?
For the part of the lab that we're talking about the cells under the microscope, do you want us to do the same thing we did for like the tubes and number and write what's happening in each tube? Or did you want us to more just like know what it looks like under the microscope?
No, I was saying you should print out these pictures, like where it says, like, look at this particular tube under the microscope. You can say, it probably looked like this picture because of such and such. It's an isotonic solution, so I expect the cells to look like this, or it's a hypertonic, so I expect this, or
Okay,
so,
thank you.
yeah, so you use those pictures. You don't have to print out every single one of them, but like have some representative cells from the different conditions and say, the cells in this tube, likely looked like this.